Yeah. So thank you very much for the invitation. It was a pleasure to come and talk to you all. So let me. I don't know how to. Yeah, I don't know how to. Yeah. Okay, perfect. Yes. So I'll talk to you a little bit. I'll be giving an overview of many of the projects in the lab. So you feel free to interrupt me with questions and don't hold them in the end if something that I'm showing is really far off from what you are used to. So one thing that one driving question that we have in the lab is to try to solve this paradox that we often see, that most diseases are tissue specific with genes that are expressed in the entire body. So I'm sure you can think of a few. In cancer is the rule and not the exception, except for 4 or 5 cancer-driving genes like p53, TET, CDKN2A. Most of the genes will be very, very tissue specific. It's not specific to cancer, but it's also true for rare disease. Oh, this Zoom is... Yeah, but I don't have the response. Yeah. Okay. It's true also for most of rare diseases. So at this point, we know about 1,000 of rare diseases where the gene is the causal gene, and you will see that 80% of those will manifest in a single tissue. So the most simple answer to this question is maybe these genes are expressed in that tissue only, and that's why you get the tissue affected. But when you look at the expression patterns of genes, they all follow the same trend. The disease-causing genes, the trait-associated genes, as well as the protein-coding genes, they're all expressed everywhere. So most of your genes will be expressed in many, many tissues. But then when they are, mutated, you will get a single organ phenotype. So the effect of these mutations will depend on context. The same mutation can be harmless or can be very dangerous depending on which cell you're looking at. And so we all know, and we all talk about context specificity, but I want to define it a little bit more accurately and more precisely. An extreme example of this is the histone genes. I know that several groups here are working on this, so you will be very familiar with these genes where the DNA is wrapped around and they have these histone tails. Oh my God! Yeah. Thank you. Yeah, no, but I lose my click. That's the problem. Yeah. So that's why I'm complaining, because it's not responding, but it's not you. It's yeah. So, you have these histone tails that have this post-translational modification that will form the histone code. And I know you have a very deep expertise here, so I'll spare you all the details on this. But they are extremely important for every single cell in the body, because we all need, all the cells need to express genes and need to have this regulatory code very well nailed down. So what happens when these mutations happen rarely in humans? It's a really interesting yin-yang. In the germline setting, you will have typically neurodevelopmental disease. So many of these, this is histone 3. Many of these positions have been reported in neurodevelopmental affected patients. And then in the somatic setting, you will have cancers, and with very, very extreme patterns. So for instance, in H3.3, if you look at position K27M and variant H3.1, you will have tumors very early in life in babies in the pons, this region that joins your brain and your spinal cord. The same position, but a different histone variant. It appeared in the midline a little bit later in age, a few residues to the right. And then you have cortical tumors, adolescent and young adults, so very striking different patterns. And then in every case with extremely dismal, so a very aggressive disease. So it's extremely aggressive phenotype. And then you take it to the extreme G34 position, depending on the exact substitution, you will have brain tumors or mesenchymal tumors. So really, perplexing patterns. We think that this is due to the cell context where the mutations occur. So the working model that we have for these particular mutations is that in the normal development of the cells, especially in the brain, that we have hundreds or thousands of different cell types, we really have a very concerted epigenetic stepwise changes determining the fate of these hundreds of thousands of cell types. And that requires the interplay of several epigenetic readers, writers, and the epigenetic state. And then you have this mutation and all this process that should be very concerted and very complex cannot move forward because we have a very strong phenotype here. And so the first step is to actually prevent this differentiation. And then that, leaves the cells in a state that, if you think about it in a progenitor or very early embryonal state, is very close to a tumor. The cells need to self-replicate, need to migrate, need to invade to just reach their final destination, etc. And so the second hit that will be typically in a tumor suppressor or a DNA repair or a signaling factor, a growth factor, is the one that actually triggers the full transformation. So that's the underlying working model that we have for these mutations. I will not claim that I invent this model. If you look at who came up with a model like this, you actually go back all the way to 1877. It was the predominant theory of cancer creation. So this is a very nice review in 1903. So before even we knew about the DNA, etc. And if you read what was one of the 2 or 3 models of cancer formation, it says that the cells became displaced from their proper relations during the process of cell differentiation in the embryo, remaining a quiescent or latent state until excited into activity by one or other or several causes, which I call the second hit, an indefinite time after birth. So of course, this doesn't explain all cancers. It doesn't explain melanoma. It doesn't explain lung cancer. So this theory was dismissed about 1925 and forgotten, and then rediscovered again for a family of tumors, which I think our brain tumors fall into that this time. So the main point here is that, according to the second aspect of this model, is that the histone mutations may hit different progenitors in the brain. And so what we think is that a large part of what the tumor phenotype has is because of the identity of those progenitors. So progenitor red here will give you one type of tumor. Progenitor blue here will give a different type of tumor. So we think the context in the cell of origin largely determines the phenotype, the molecular phenotype of the cancer. And we think that because cell identity programs and a lot of what the cell is, let's call it identity programs, are extremely strong, evolutionary and conserved. So this is, I give you a couple of examples. So this is an old result that has been replicated 1 million times. If you take RNA-seq samples from different species and different organs, they will first separate by organ and then by species. So here you have all the brains and here you have all your, heart. So in other words in expression space, your brain is closer to a chicken brain than it is to your own heart. So it's really that evolutionary conserved. So this is, supposed to be sustaining many, many years in evolution. This is another example of robustness of these type of programs. Here we have 16 regions of the brain. In the x-axis, we have age. So from 8 weeks in the womb to 40 years of age, life, so 40 years. And you see one gene, how defined the pattern is across all the different regions of the brain. You take CBX2, a very related gene, CBX7, and you will see how the opposite pattern is. So it's the same complex, PRC complex, and you see how tightly regulated this is. So these are extremely strongly regulated and extremely robust type of programs. I don't think they will be perturbed by a mere transformation if they are designed to sustain, millions of years of evolution. So we think of those as fingerprints of the domain of origin. So I was born, everybody was asking me, in Argentina. I spent my life in Italy, then in Mexico, went back to Argentina. Then I came here 20 years ago. I became Canadian citizen. If you look at my wallet, you will see my, Quebec transit, a lot of papers. If you ask me where I'm from, I don't even know anymore. But if you look at my birth certificate, it didn't change. So we think that there are some programs engraved in the cells that don't change no matter where the cell goes, etc. So that's the underlying principle of what we do. So the strategy we have been taking in the lab is now we go back to the normal developmental brain in our case, because we look at brain tumors. We take normal developmental references at the single cell level, very large. We use them to analyze the tumor heterogeneity. The most important is for each tumor sample, each tumor cell, we map them to these references. We find the closest match. And once we find this closest match, that's where we turn and try to understand why each mutation is associated with a specific cell type or a specific progenitor. And then we do all other data modalities and we do modeling, etc. So the strategy kind of worked quite well. So if we look at, how do we map? I didn't put, million slides on the computational aspects, but I'm happy to answer questions. I think I showed a few in the meetings today when they were asking me how we do it. But in a very high level, we do two types of approaches. One is signature enrichment. From these references, we take cell type signatures from each cell type, so from hundreds or thousands of cell types, and we score those cell identity signatures in the tumors. And the second is the data sets and the references are so large that we can train machine learning classifiers in each one of these references. And then we use them to predict each tumor cell, what is the predominant lineage program that it is. So two major families of methods. So if we look at, the variety of pediatric cancer, so this is a profiling of 1,000 cancers a few years ago, not by us, that classified tumors in different, groups. We kind of mapped several of those already to very, very distinct progenitor states, progenitors that are anatomically very different in the brain, so really different regions. And this is not only for histone mutant tumors like these two, but also for others that are not histone mutants. So I think it's more general than just the histone or epigenomic mutations in the brain, at least, that we have this diversity. Really, each tumor type will be associated with a specific, very, very tightly associated with a specific cell of origin. So let me tell you a few stories about why this association with a specific lineage and a specific mutation, what did we learn about the tumor and what did we do about that? So the first one is these G34 mutations that, if you remember, they are causing, cortical tumors in adolescent and young adults. Under the microscope, they have like a dual phenotype, is neurons and glia in the same type of tumor. So kind of, very bizarre and chaotic. So a few years ago, Nada Jabado and Carol Shen, who now has her own lab in British Columbia, assembled a cohort of about 95 patients. And we saw that half of those had mutations in an extracellular domain of PDGFRA, is a signaling receptor. And the ones that were not mutated, most of them also overexpressed this gene. So in this case, the partner mutation, the second hit, was PDGFRA, so this second hit. In parallel, Celine in my lab was mapping these tumors and found that these tumors were mapping very precisely to specific progenitors in the brain. So each column here, it will be one cell type. And so these tumors were having a strong signal for radial glia, which is the stem cells in the brain, neuronal progenitors, prenatal interneuron inhibitory, but not the postnatal and not any other type of neuron and not glial. So very, very early neuronal progenitors of inhibitory ones. So in our mapping, we have a second hit and we have a specific lineage. So why we have that? So, well, the cortical neurons, so cortical are all the ones that are here. They are actually born in one specific domain in the ventral brain, and then they migrate, okay? And the way these cells are produced in the embryo, they migrate, and then they will be your inhibitory neurons for the rest of your life in the cortex. It's quite well defined, so these progenitors need to decide between two fates, interneuron or oligodendrocytes. And the set of transcription factors that determines this fate decision is very well established in the neuroscience literature. So you have these three transcription factors, GSX2, DLX1, and DLX2, will determine this fate and will repress the alternative fate. And those are the transcription factors that are really active in our tumors. So they are really mapping to this particular cell fate decision. And so we can complete, our schematics here. And so we have the interneuron fate, G34 first, and then the second hit, PDGFRA. The question is why? So here is where it becomes very interesting. You have to think about this as at organism level. So this progenitor domain needs to produce all your interneurons, so the thousands of interneurons that are here, and also some oligodendrocytes, and it has to produce them in a very coordinated way. Because if what happens is that this domain will produce the two cell types and they migrate together with the oligodendrocytes guiding the neurons. So this same domain needs to produce the two at the same time in the right amounts. If you remove this because you do genetic manipulations, all your interneurons get lost and never reach the proper place. So they really need to be coordinated. So how does your body coordinate the production of two different cell types in the same region? Well, oftentimes it does it with chromatin regulation. When we look at where these two genes, the one that determine the two fates, are in the genome, well, they're next to each other. When we saw that, we saw, what's happened? So that was a really a moment of surprise. What is happening here? So that's where we started speculating. Well, maybe these two are actually co-regulated, and maybe they are actually in the same 3D domain on the chromatin, so that the cell can choose between the two fates. So we did a high C chromatin conformation, which looks at the 3D genome distance between the genes. And we saw that in the normal setting where we were proposing that these tumors arise, and I put the drawing here for the people that are not familiar with high C, what we have is a structure that puts together our oncogene with the lineage transcription factor and the active regulatory elements here, which are this triangle here. So this is happening in the normal setting for a really limited amount of time while these neurons are produced. And then we can quantify this. And in the normal setting, this oncogene is actually repressed by histone marks. So the things are close and the injury close, but it's not expressed. What's happening in the tumors is that they actually steal this conformation. And because we have a histone mutation, now we have the overexpression of the oncogene. So what we have is the same situation, but now our PDGFRA gene gets expressed because it's close to an enhancer, an active enhancer. So that explains a little bit why we have the second hit, PDGFRA, only in the interneuron lineage. Well, this region is only active in that lineage. So that's why you get the opportunity to get your tumor. Why we have the mutation actually perturbing this regulation, we don't know yet. So that's the holes that we need to actually fill. Why exactly G34? So we know why the interneuron and PDGFRA, why G34. Some hints we got when introducing, again, with Nader Jabada, the mutations in the germline setting, and we saw that the lineage that was most affected was the neuronal lineage. So we saw reduction of the neurons in the brain, and that was affecting every single neuron in the brain. So it was preferentially affecting the neuronal lineage. We still don't know exactly why, but we think that is, I won't spend too much time here. It's just to tell you that we have some clues, but we think it's because the mutation is affecting a DNA methylation process that is really affecting only the neurons. So, but I want to go back to the cancer setting. So this was a parenthesis. So what do we do with that knowledge? Well, we can go to our friends modelers. So we work a lot with Manav Patania in Cambridge that produces mouse models, and that does that by delivering the mutations in utero in the specific places in the brain, and then we get the tumors later. So we said these G34 tumors originate in the ventral brain. So can you try? And if in effect, delivering the mutations in the dorsal part gave no tumors. when you put it in the ventral part, you get tumors, really nice tumors postnatally as predicted. So at least we know that this specific progenitor niche is capable of forming the tumors. So to summarize this part of the story, which is the G34, is a story 50% of those have a mutation in a second oncogene that we think they originate in this interneuron lineage. These neuronal lineages are particularly sensitive to the G34 mutations. And the 3D genome in this cell of origin is actually favoring the co-option of the oncogene, and that explains why we see this association in the patients. So that's for the G34, a story, and it's all paper, so that's why I made it very, very short. What happens with the K27N mutation? So K27N mutation can hit two different histone variants, H3.1 or H3.3, and as I said, they will give you different locations, but also different association with partner mutations. So H3.3, you will find it either with p53 and ATRX, or with NF1 or PPM1D or PDGFRA. So again, signaling, DNA repair, tumor suppressors that actually are the ones we think are promoting the full-blown transformation. Here in the PONS, H3.1, most of them are associated with ACVR1. So why, you see? So I'll tell you what we found and why why this association in particular. If you ask me, like many people ask me, why the others, I don't know yet. We're trying to figure them out. But we have some answers for this particular association, why the second partner association. So regardless of the differences between H3.1 and H3.3, the position on the genome, etc., partner associations, the convergent effect of these two mutations is to restrict K27ME3. So when you have a K27N mutation, the repressive mark K27ME3 gets collapsed and only restricted to the landing sites of PRC2, so CPG islands. And if you remove the mutation, the mark will spread. So if we go back to our fingerprints, what happens with gene programs that will determine your anatomical or positional or cell identity? So let me show you one of these examples. The Hox genes. Hox genes are clusters of genes that will tell the cells where you are. If you're looking at your anterior posterior axis in the body, it will tell you where you are if you are in the head or if you're in the spinal cord, and exactly where in this axis. The way they do that is a cluster of genes. So it's 4 clusters of genes that have a very defined chromatin conformation, and that is actually co-regulated with the identity. So the cluster, so here is an example of one cluster of genes where you have all the Hox genes, and you have 2 domains here. I hope you can see them. One half of the domain is active, express, and the other half is inactive, repress, and linked by chromatin loops with CTCF. And so the position exactly of this boundary between the 2 domains will tell the body where you are positioned in the body. So you have really, really beautiful example of fundamental biology, exquisitely preserved. And that is also accompanied by, chromatin conformation that determines the boundary of the 2 domains. One essential part here is K27ME3. And I told you that was completely disrupted in our tumors. So the question is, what do these clusters look like in our tumors? Would that have this massive epigenomic disruption? So if this is the normal setting, look at how beautifully organized it is in our tumors, including, a lot of the position of K27ME3, the presence of the 2 subdomains. It's beautifully organized, and it points exactly at where the tumor is coming in this axis. So it's preserved. It's not disrupted by transformation, and we can use it like to, as a birth certificate where these tumors were specified, the cells. A second set of transcripts of patterning genes or transcription factors that are positional identity are these 2. So Hox genes will tell us position here. PAX3 and NK6.1 tell us the position in this direction, anterior, posterior. So when we're closing the neural tube in the development, this is the dorsal and ventral setting. We have gradients of BMPs and sonic hedgehog that will determine the expression of these patterning genes that will give us, discrete domains of progenitors that will specify all our cells later. So there's one parenthesis I want to make for the non-neuroscientists here is that we have cells that, will live with us for the entire life. So let's say OPCs, oligodendrocyte progenitor cells, we have them all our life, but they are specified in the embryo, the same as, our eggs and gonads are specified in the embryo and then they stay specified for the rest of the life. It's the same for most of the neurons and for the OPCs and many of the glial types. So we have them, they self-renew, they proliferate, but they are specified in very specific waves like that. And they retain a lot of the characteristics for decades, right? So these two genes are that we were finding in H3.1 versus H3.3 were actually pointing us to where the cells were specified. So H3.1 tumors were NK6 positive, PAX3 negative. That pointed us to a very specific domain given the Hox configuration and these patterning genes in the ventral domain here specified by NK6.1. So it seems very complicated, but why I'm telling you this? Because this specification is extremely robust. So dorsal cells specified by BMPs were born seeing BMP. They have internal mechanisms to deal with BMP changes because that's how they are programmed. This is one of the most beautiful computational biology papers of 20-something years ago, predicting the mechanism of how this, gradient is dealt with these cells. And it's actually verified many years. We verified it in our data. I don't think I have the slides for that, but this is, the cells that are specified by BMP will continue to respond to BMP for the rest of their life, and vice versa for sonic hedgehog. So super robustly specified. So the point is here that if we have a mutation that increases BMP signaling, these cells, well, they know how to deal with that. That's what they are born how to do. What happens if we put a mutation increasing BMP in a cell that never saw BMP and that has no mechanism for dealing with this? Well, we're in trouble. So our H3.1 mutations are specified here. They are not wired by BMP. So when we introduce a mutation that is inducing BMP, then you have an oncogenic phenotype. So the reviewers were asking us, well, show us that, this dependence on BMP is real. So they actually maintain the vulnerability. If we remove BMP, you will decrease the downstream phosphorylation, phosphosmad of ACVR1. We decrease downstream expression of the effectors. We decrease the capacity of colony formation in vitro, and then we decrease the capacity of tumor formation in vivo. So they really are needing this BMP to proliferate, and this is wiring who they are. And so another parenthesis to say that this anatomical identity is not something specific of our weird tumors. It's been found again and again, especially later. So this is in melanoma, similar concepts where regulatory networks of the cells that you have in the palm of your hands versus the other cells will tell you whether a mutation will cause a tumor in one place or the other. Similar for kidney cancer, similar to lung cancer, so very heterogeneous adult cancer, similar principles apply. And so the common theme here, and that's why my original image in the beginning, is that we need these cell fate specifications, some characteristics that are a double-edged sword. So here the need to coordinate these two fates makes it so that you have an oncogene very close to a very active regulatory element, and sometimes it goes wrong and you have a tumor. And that's the price you have to pay for specifying properly your cancers. Same story for the K27M mutation. They need to have different progenitors and OPCs responding to different signals. Well, put some in danger of specific mutations because you have them so robustly specified that they don't have flexibility. These ones are BMP, these one are sonic hedgehog, and there's some robustness there that is determining why we have the tumors. And I'm good on time, right? For 15 minutes? Mm-hmm. Perfect, because I want to tell you the third story that is not histone mutation, which is another tumor that we also map very closely to another ventral progenitor like this one. So FOXR2 neuroblastoma are another tumor similar to the G34 that under the microscope, they have this dual phenotype of oligo and glial and neuronal identity. So they are really mixed, messy, we would say. If you're a pathologist, I don't remember the good word for messy, but they are messy, they get misdiagnosed. And FOXR2 is a very potent oncogene. So it's a pan-cancer oncogene that will create tumors all over the place. But if you combine in the brain FOXR2 neuroblastoma, they will form a very, very concise entity. So we decided to look at them as a separate entity because they have a very close, very concise methylation pattern. And we tried to identify the origins of this and see if we could explain this messiness phenotype under the microscope. So what are they? Are they glial? Are they neuronal? Do we care? Is just abnormal differentiation? Is just abnormal cancer programs? Or can we put some order on it? So what we did is we collected, as usual, a large number of patients. So FOXR2 is a very rare tumor, so we got 28 samples. And then we use other tumors that are also neuronal, like medulloblastoma or ETMRs, and some glial tumors as comparators. So high-grade glioma, PFA ependymoma, so that we can compare them to different entities and similar ages and similar locations. So we had a nice cohort to compare to. So if we learn something over the years about these fingerprints, we said, can we systematize it? Can we show that these fingerprints exist? Can we derive them? So can we find them? So what do I mean by, can we formalize a little bit better? What do we mean by fingerprint of anatomical domain of origin? Well, what we mean is a combination of transcription factors that in the fetal brain are discriminant of different anatomical domains. So they can distinguish different anatomical regions, that they are sufficient for us to infer where the cells come from, and that they are maintained through differentiation migration, from prenatal to adults. So this is an example of what I mean. So these are 3 transcription factors that in the brain, this is RNA and this is a protein level. So TBR1 will mark this dorsal part of the brain, NK6.1, this ventral domain, and this one, this other. So we derive 14 of those from literature that we verify in these atlases of the brain that they are marking different domains and that we think are sufficient. So the question is, are they sufficient? So what we did to verify whether they are sufficient is we took references of the normal brain from the neuroscience community. So there's the Allen Brain Atlas. So there are several, large consortia initiatives that have really large data set annotated. So 5 different references, human, mouse, fetal, and adult, about 500 cell types that are annotated by their origins. And so we train on support vector machine, but only on these 14 genes. And then we ask, can we tell us where the cells come from? And a cross-validation experiment, the sensitivity was 96 to 100%. So yes, they are sufficient. And then we remove a few that we couldn't detect. So this is what it looks like. So these are summaries of single cell references. Again, human, mouse, fetal, adult, and you can see how different, they are preserved across the lifespan. So the idea is that if they are so preserved, mouse and human, and all the way to adulthood, they're likely preserved in after transformation. So we map our tumors, and the FOXR2 we're mapping to this medial ganglionic eminence derived, not the other tumor types. So the G34 that I told you were derived from this LGE, they map the LGE, etc. So by this analysis, they were mapping to the MGE. Of course, these are only 14 transcription factors, so they could be induced by FOXR2. How do we know they are not? It's just a few genes. So this is what these 14 transcription factors look like in our tumors. And this is what they look like in another tumor type, the APG, that also has FOXR2. They are not induced in another tumor types. If we look in human neural stem cells and we put FOXR2, they are not induced, so they are not downstream of FOXR2. they're likely a memory of the cell of origin. So that's, again, just 14 genes and a gene signature. We also did the gene signature enrichment, like unbiased, and also the machine learning approach. And this is single cell data from tumors, and predominantly they were mapping to this MGE. So the mapping was quite clear. So with this mapping, and this is a rare tumor, so typically modeling rare tumors, modeling any tumor is very expensive, and you know, you're all PhD students, many of you are PhD students, and you know how long it takes to create a model, sometimes many years, and you don't have any phenotype. But here we have a lot of information. So we can go to our friend Manav and tell him, please inject these specifically in this region. And in the first try, he got the tumors with 50% penetrance, and they really looked like the human tumors with the dual neuronal and glial phenotype. So it was a very fast way of modeling the tumors with, you know, previous knowledge, using references that are already there. So, you know, people have spent hundreds of millions of dollars creating these references, but for us, it's free, so we can use it. So now that we have the models, we have the mouse models, we can start asking the question, can we uncouple what is cell of origin versus downstream effect of FOXR2? Can we uncouple all the signals? So from the mouse, we did first single cell multiome, which is single cell profiling of RNA and chromatin accessibility. And he also derived cell lines. And from the cell lines, we can do some cut and run for FOXR2, so to know where the oncogene is actually binding. So the first thing I want to show you is the intersection between the FOXR2 here on the cell lines and the multiome in the mouse. So we have a pretty good overlap that is telling us three things. The first one is that the cell lines are pretty much recapitulating the chromatin landscape of the mouse tumors, because we have a very, very good overlap here. So that's very reassuring. The second is that FOXR2 antibody is not so unspecific, so most of it is binding in open peaks of chromatin and not background. And the third one is what we know, but it's really nice to see in, you know, in numbers, is that FOXR2 or the oncogene only binds what it combines, which is what is open in that particular cell type. You see, so there's very few peaks outside of what we see as open chromatin here. So my point here is that the oncogenes, even if it's a pan-cancer oncogene like FOXR2, the downstream effects will really depend on what is accessible to that oncogene, which is this space of open chromatin. So we did a motif enrichment for FOXR2 to see where it was binding. And, you know, in my 15 years of bioinformatician, I don't remember seeing such a clear signal. So the peaks that were, you know, the motifs of transcription factors in FOXR2 peaks were actually two families. Those are the top ones. I didn't choose them. Is ETS and SOX9, SOX10, and FOS. So those are two different signals: initiation of proliferation and early gliogenesis, right? So this is what the chromatin conformation around one of these look like. So this is one of, this is ETS1, one of these genes. You will see FOXR2 binding all around close to this. You will see the gene being expressed. And then in the multiome data, we have the ATAC data, the chromatin accessibility separated by the type of tumor cells that we have. We have it open in the right tumor cells. So everything is very consistent with FOXR2 helping express these genes unbinding. And the same goes for SOX10 and SOX9, and I will not show you we looked at all of those and they are really consistent with oncogene binding, opening chromatin, and expressing. And the genes SOX9 and SOX10, together with 3 or 4 more transcription factors that I don't show you here, but they have the same patterns, are actually sufficient to initiate gliogenesis, even when you have fibroblasts in culture. You know, when you have iPSC, etc., you can convert fibroblasts to glia using these 5 transcription factors. And the same goes for all the gliogenesis pathways. So very, very consistent. So the model that it helps us build here is that on the background, on a very, very robustly specified neuronal fate, which is typically very robust, you have an oncogene appearing. And in this open epigenome, you have activation of initiation of proliferation and gliogenesis. So the messy phenotype that we see under the microscope is actually secondary. The secondary acquired phenotype is the glial, the original is the neuronal, and then probably what is happening and what is relevant for the cancer biology is all this ETS and proliferative, and the rest is, somebody was asking me today, do you care more about lineage? Do you care? Well, it depends. In this case, I think the gliogenic phenotype may or may not be relevant for the cancer for us to target. Is probably these ones that we want to stop targeting, etc. But at least we make sense on the association of this particular tumor type with these 2 or 3 mutations that we see. So to conclude and to go across the 3 stories that I told you about, is in each case we have really massive disruption, either by histone mutations or by a very, very potent oncogene. And in all cases, the key developmental cell identity programs are still very, very recognizable, and we can fish them out and we can use them to understand the tumor biology. That each pair of mutations will associate with a very ontogenetically, meaning developmentally different progenitor with very different characteristics, and they maintain their vulnerability. And that if we leverage these single cell references and this normal knowledge, then we have a very cost-effective approach to model, to guide the rational creation of models. And with that, I will thank a lot my lab. These are the 2 people behind the FOXR2 paper. Celine was, I think I mentioned her sometime in one of the meetings. I needed 5 new students to replace her. That's her face. She's amazing. And she was behind all the stories I told you today. And then everything is done in super close collaboration with the lab of Nada Javado and Makiel. So private replication very abundant. What happens in the plants is that in the first few years of life, you have a massive expansion. So that's, I think in that case, that's why we see it at that point, because you have like, I think it's 100 times, and it's really, 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 really massive expansion in a very short time. So that's a canonical, very abundant. You really need the cells to be replicating to see the effect. So I think that's the timing explains that. Then there are you have the great science that shows how the interaction figure. So we look at the specification, but we're not certain about the planning analysis. So I think for the planning,
question about this loops, chromatin loops. So the only H3.3 mutation which drop this loop, but not canonical, right?
Yeah.
Like, do you know why? Is it because some nucleosomes with H3.3 actually deposited somehow in the vicinity of this interactions?
Absolutely no idea. Yes, no. Yeah, I was wondering the same. Yeah, and I don't have a good answer yet. No.
In another question, so you like your model explains this this loop explains like you mentioned 50% of patients have this mutations in H3 G and this another gene. What about other 50%?
So 50% have the mutation, 20 or 30 more are overexpressing the gene, so it will be the same oncogenic mechanism. Then we have 20% that are not explained by this. Some of those are not mapping to the interneuron. So there may be some G34 mutations that are not following this. So yes, we're looking into that, but 20% of rare tumors is really hard to get the samples. But yes, yeah, it's not universal. Yeah. Yes, of course, it's been affected. So most of the mutations are extracellular domain mutation, which is a whole other super interesting side that we are studying. So extracellular mutations are, so you have, pediatric, you have the kinase domain mutation. That's an extremely potent oncogene. So that will give you gastric cancer. It will give you, whenever you put it in 2 weeks, you have a mouse with a tumor. The extracellular domain are all be found in the brain. So why? You see, so it's an environmental thing in microenvironment. So that we're looking, but it's a very difficult problem. There are some experiments with kinase inhibitors, and typically the genes, the tumors will become resistant in no time. There will be another kinase treatment. Yeah. So I don't think a direct inhibition of that VDFR is a viable.
That's really nice, thank you. So I understand how the cell of origin kind of helps explain, and it's so beautiful that you can then pinpoint where to put the cancer cells, because that's where you're going to see an effect. But as you mentioned nicely at the beginning, some of these mutations are acquired. They're somatic. So what I'm still struggling to understand is how are they acquired in specific cell types rather than others, or are they being acquired everywhere?
So
and you only see the effect in those tissues.
So this will come into the speculation.
Yeah, that's good. I want speculation.
Exactly. So I will speak because I have a lot of speculation there. So first, in the models that we do with MANAF, those are actually, we're not putting cancer cells, we're putting the mutations, and we put them in it's a transfection system that is mosaic. Okay. So that's why the mice can survive, because only a few cells will, so it mimics the somatic cell. It will be only a few cells that will actually, so it's kind of modeling the somatic aspect. To your question of why the mutation, so I think even for many of these, especially the histone mutations, K27N, etc., I think the rarity, I think it's in line with what you would get by chance. Okay, I think. The second, and that's my question that I have never have an answer is, so if we model these tumors, we got to this intrauterine system because before we tried to, when I say we, it's my collaborators, right? They just do the data analysis. But if you put them too early, it's lethal. So imagine how many things this want. Same goes for the Swiss SNF complex, SMARCA1, SMARCA4. You put them too early, it's really lethal. You put them too late, either nothing happens or you get leukemias, so you don't get intracranial tumors. you really need this window of development, right? So I don't think necessarily the mutations happen there, but I think they are tolerated there for some reason. So I think it's a survivorship bias for the very deleterious of K27M is I don't know why these progenitors can sustain them and not the other progenitors. For the more milder, so G34 is milder, the effect, so it may be not, but the ones that are really, but you never see it, you see, I think it's a survivorship, is tolerance to the mutation, and I don't know why.
And overall mutation burden is very low, yeah?
Sorry?
Mutation burden overall.
It's quite, well, it's a variable depending if they have P53 or if they,
Yeah,
you
okay.
have some patients that are, but mostly in general, as an average, is very low. Is very early, and when we put them in mouse, it's like you really need 1 or 2 hits and you get a full blown, very aggressive too. Yes, they're really sufficient.
Any other questions?
Yeah.
Students? Okay, thank you so much, Claudia.